All right, guys, let's dig in here and really start to get a look at how this chemistry of life really sort of gets going, right? And um, get into a discussion here about the steps of this really key pathway, glycolysis. Now, as you'll recall, I mentioned last time, the first few reactions of glycolysis are this energy investment phase, right? We're going to have to put in some ATP to do what I call prime the pump. And we're going to take glucose and we're going to make this diphosphorylated version of it, fructose 1,6-bisphosphate, right? And to do that, we need three basic reactions to get this to go, right? Okay, so let's just start at the top here with the very first reaction, reaction one. Where we're going to take glucose, right? And we're going to take some ATP. We're going to phosphorylate one of those alcohol groups on the glucose to create glucose 6-phosphate, which if you think back to the beginning of the course and all through the course, I've been using this as sort of a through line all the way up to this point in the course. We've talked about this reaction several times, right? And I'm going to use the symbol, that P with a little circle around it, to indicate inorganic phosphate, right? Or a phosphate group that is linked through an ester bond, a phosphoester bond there, right? Okay, so finish drawing out my glucose here, right? So plus ADP, right? So as I said, we've basically done a group transfer reaction from ATP, that phosphodiester, to create this phosphoester on that alcohol group on the 6 carbon, right? Just as a reminder, you guys, the nomenclature here, right? There's our 1 carbon, 2, 3, 4, 5, and 6, right? So this is, of course, taking, as I said, glucose and making glucose 6-phosphate, right? All right, or sometimes it'd be abbreviated. And I'll probably do this to some extent with some of these, G6P for glucose 6-phosphate. Okay, the enzyme, of course, that runs this reaction, as you guys all know now, because we've talked about it a lot, is of course hexokinase, or also HK, right? In shorthand, right? And the hydrolysis free energy for this reaction, the standard state free energy for this reaction, is in the range of -17 and -16.7 kilojoules per mole, which is quite favorable. But in a living cell, the sort of under sort of cellular conditions, in most tissues, it's going to vary somewhat depending upon metabolic state and tissue type, but it's in the range of -33 kilojoules per mole. So this is an extremely favorable reaction sitting right at the entry point into glycolysis, right? This hexokinase reaction. Now, hexokinase is an enzyme. It's probably worth knowing. HK is pretty promiscuous. So this would fall into that category of enzymes where you have multiple substrates, right? And the Kcat or Km for a substrate is going to depend upon, which enzyme, which substrate gets used under a given set of conditions is going to depend on the Kcat or Km for that particular substrate, right? So hexokinase can phosphorylate a lot of different hexoses for the purposes of running them into glycolysis or for other purposes, right? So it'll work on a lot of different hexo sugars, but glucose is one of the main sugars that's going to go down this pathway. So it's one of its big substrates. There's a related enzyme in liver tissue called glucokinase. right? And liver's a little special, right? We're going to talk about this a little bit. In addition to just having to be a living tissue and run some of these metabolic pathways, liver plays this huge role in regulating blood glucose levels. And so glucokinase in some of the key tissues or cell types in liver tissue has a much, much higher Km than hexokinase. And glucokinase, therefore, can respond to much wider changes in glucose concentration level and not have, it also tends to not be inhibited by the product, glucose 6-phosphate. So it can respond to these big changes in glucose level, which is important for what liver tissue does. But for most other tissues, hexokinase is the key enzyme here at the start of glycolysis. Hexokinase also in most of these tissues spends most of its time operating very close to its Vmax because in most cases, glucose, unless the cell's starving to death and has run out of its glucose stores, the concentration of glucose is so high relative to the HK Km that this enzyme is usually running really close to its Vmax, right? So it's generally when tissues are running glycolytically, hexokinase is kind of running flat out under most circumstances. All right, so let's think a little bit about the role that what we're doing here and why. So we phosphorylated the sugar. Why are we doing that right at the beginning? I don't know if you guys remember or not. There are 2 or 3 different reasons why we're putting phosphate onto the sugar, right? One is that if you look at glucose, right, remember this molecule, glucose, this is what we want to yank apart. We want to suck electrons out of the structure of this molecule in order to make ATP. But this molecule is reasonably polar, but it can also diffuse its way through membranes, right? Not, it doesn't just come flying out, but it can diffuse through a membrane. By phosphorylating the sugar with this hard negative phosphate, you dramatically inhibit that. You essentially trap the glucose inside the cell. Once it's been phosphorylated, it's not going to get back out again, right? At least not very easily. In addition, we're going to put multiple phosphates. We'll put 2 phosphates in the sugar ultimately, and then we're going to essentially end up concentrating a whole bunch of negative charge around the structure of this Pyranose ring, right? We're going to change the structure in a minute. That concentrated negative charge is going to help provide a nice enthalpic driving force for splitting the sugar right here between this 3 and 4 carbon. That's ultimately what we're going to go do, right? All right, and having that negative charge on sort of 2 halves of the molecule helps provide an enthalpic driving force for helping assist in separating bonds. So you can think about this phosphorylation as sort of a destabilizing condition that helps make the splitting easier. The third reason that we're putting phosphates on the sugar is because we're going to use them in a key step of in the glycolytic pathway to directly make ATP. Some of the phosphates we're going to put on here are going to go directly into creating ATP from ADP in a direct substrate level synthesis, right? Okay, let's talk a little bit more about hexokinase as an enzyme, right? Just to refresh your memory, right? You remember this enzyme has this sort of Pac-Man-like structure, right? It's got these 2 lobes. It's similar to the actin fold. And you remember, of course, that when glucose binds into the active site of the enzyme, the Pac-Man kind of closes down. And we just talked about in lecture a couple of lectures ago, the importance of these conformational changes in enzymes as they recognize their substrate, both for the purposes of positioning key residues, for stabilizing the transition state, for straining the substrate molecule, and that this conformational change, this induced fit idea, is such an important idea in the in basic, enzymology, right? The idea that enzymes are not static structures, that they change their conformation is a really key one. And in addition to getting this the substrate ready to react, the conformational change in hexokinase plays one additional role. If you think about it, what's the enzyme about to do to the ATP, right? We want to do a group transfer. We want to take a phosphate group off of that ATP, that energetic that energetically unstable, high transfer potential phosphate group. We want to stick it over here on glucose, right? And we're going to use that alcohol group on glucose, going to activate it. It's going to be a good nucleophile. It's a good place to make a phosphoester. There's something else hanging out in solution here that would be really wonderful at doing that too. And there's a lot of it, really high concentration, right? Water, right? There's a lot of water, right? As we've talked about before. So imagine what would happen if that ATP group, that ATP was in the active site of the enzyme and there was water hanging around, and we're going to start positioning some residues to stabilize that high negative charge transition state we talked about a few lectures ago. And instead of a glucose, that alcohol group from the 6-carbon glucose sitting there ready to attack the phosphodiester, there was a water molecule. That would just waste the ATP, right? You've basically blown it, right? So in addition to positioning residues for effective catalysis, getting these two substrate molecules at high concentration where they need to be, all the things we've talked about for catalysis, another important role in this case is for this conformational change as it clamps down around the substrate to eliminate water, dehydrate the active site, eliminate water from the active site, right, and from the substrates, so that the only way in which the activated ATP can react is with that glucose hydroxyl, right? Okay. All right. That's reaction 1, hexokinase, right? We have introduced that to you guys from several different angles. Hopefully you're going to remember that guy. All right. So let's go back now and think about the second reaction. So we've made our first substrate, glycolysis, glucose 6-phosphate. Now let's talk about the second reaction, reaction 2. Now we're going to take that glucose 6-phosphate, right? There's our, whoops, let's not be so sloppy. There's our glucose 6-phosphate. All right, and now what we need to do is ultimately, we want to get to the point of having this sugar with 2 phosphates on it, right? And we want to put the other phosphate on the 1-carbon. So we want to basically have a molecule that's symmetric with phosphates. One on, it's 6 carbons. We have one on the 1-carbon, one on the 6-carbon. So now you can imagine, say, we're going to cut it in half, sort of be symmetric, right? Okay, but to do that, we've got to move the carbonyl. Remember, this is our number 1 carbon here. Glucose, if you recall, is an aldose sugar, right? There's our number 1 carbon. That oxygen, which is what we want to put the phosphate on, is tied up in this internal hemiacetal, right? If we phosphorylated that, and I don't remember off the top of my memory, though, is that acetal oxygen is much less nucleophilic than a standard alcohol. So that even if you can get this to go, it's not going to go well. But the bigger problem is if you did that, you'll trap that molecule in that Pyrenos ring. You can't do the ring opening you need to do to ultimately split it. So it would be dead, right? So to prevent that from happening and have glycolysis work the way it needs to, we need to move that carbonyl group over. And that's what this second enzyme is designed to do. Okay, so we're going to come in here and we're going to take glucose 6-phosphate and we're going to turn it into fructose 6-phosphate. Right, there's our 6-carbon, our number 6-carbon. Here, there's our phosphate group, right? Now, we've liberated that hydroxyl that was on the 1-carbon because we've now moved the carbonyl over to the 2-carbon, right? To basically turn glucose 6-phosphate into fructose 6-phosphate, right? Or F6P. And this is G6P, right? Makes some sense, right? So essentially, we just turned an aldose into a ketose. And if you remember from earlier in this lecture, the kind of reaction that does that is, of course, a tautomerization. So the enzyme that runs this reaction, known as phosphoglucoisomerase, it's a mouthful, or more simply, PGI, right, is essentially an aldose ketose um, isomerase, right, or tautomerase, right? So we're just going to run in this conversion. What this enzyme's doing is running a tautomerization reaction, right? Okay. And the free energy for running this reaction, or the free energy balance, this is interesting. All right. There's an interesting little wrinkle here. Um, the standard state free energy of the PGI reaction is plus 1.7 kilojoules per mole. Let that sink in for a second. This is the second step of glycolysis, of this essential pathway. We don't run this, we die, right? Any cell-based organism on this planet, as far as I know anyway, they all depend upon this pathway at some level. And I just said the second step is thermodynamically unfavorable, right? As written, it's much more favorable for PGI to take fructose 6-phosphate and turn it into glucose 6-phosphate, right? The aldose is more stable than the ketose form of this enzyme, of this substrate. So what gives? What do you think happens? Well, if you remember, right, equilibria can be moved around based upon changes in the concentration of the reactants or the substrates. And it turns out that under cellular conditions, the concentration of fructose 6-phosphate is kept so low or sufficiently low that the free energy change under cellular conditions for this reaction, remember, under cellular conditions, is kept low enough that the free energy balance is actually minus 2.5 kilojoules per mole, right? So you can think about the subsequent steps we're going to talk about are basically draining this fructose 6 pool to pull this reaction in this direction, right? That's pretty cool. And you're going to see some more examples of that as we go along, right? Now, in terms of thinking about what's going on here, the basic chemistry of this enzyme is relatively straightforward, except it's got some extra steps, right? The first is you can't do this tautomerization until and unless you open the ring, right? So you have to disrupt that internal hemiacetal. And the way that's going to work is through some coupled acid-base catalysis, right? Essentially, you're going to have a base here that's going to yank that proton, right? Have these electrons flow back into that carbonyl, split that acetyl bond, and we have over here a nice acid that can donate a proton, reprotonate that carbon 5 alcohol, and we've opened the ring, right? It turns out similar mechanism, of course, is going to be used to do the tautomerization reaction. We're going to see a little more detail of this. We're going to do another one of these tautomerizations in a few more steps here, and I'll show you in a little more detail how that's going to happen, right? So, but for the PGI mechanism, you got to open the ring first, run the tautomerization, then reclose the ring into the furanose structure, that 5-membered ring structure, right? And now, of course, we have created our available hydroxyl group, right? We can now go to the next step of glycolysis, reaction 3. Right? Where we're going to take that fructose 6-phosphate that we just made. Whoops. Oh. Fructose 6-phosphate. Right. We're going to burn a little more ATP. This is the end of our energy investment phase. to phosphorylate that um, uh, number one carbon. Remember again, right? Here's our number one carbon right here. Here's our 6, right? So we're now going to turn around. We're going to phosphorylate that guy by using some more ATP, another group transfer reaction here, to generate this diphosphorylated fructose. Fructose 1,6 bisphosphate, right? Or F1,6BP, right? Free energy change, or I should say the enzyme that runs this reaction, of course, is a famous enzyme. It's known as phosphofructokinase, or PFK for short. The standard state free energy change for the PFK reaction is, of course, quite favorable. We're burning some ATP, right? Delta G naught prime is minus 14.2 kilojoules per mole. But under cellular conditions, it's quite a bit more favorable than that. It's actually minus 22 kilojoules per mole, right? Which gives you some sense of what's happening to that fructose 1,6 bisphosphate pool, right? When the cells or tissues are running this pathway glycolytically, this pool, right, is getting drained, right? It's getting pulled away. In fact, it's worth noting that the PFK reaction is so highly exergonic, and the pathway is so at this point has expended so much energy. By the time we get to the point of burning 2 ATPs to get to PFK, if the pathway is going to run this far, this sugar is committed at this point. Once the sugar gets turned into fructose 1,6 bisphosphate, it's more or less committed, right? It's gonna go down the rest of the pathway. It's basically downhill from this point on, right? So that makes the PFK reaction this sort of major committed step, right, in glycolysis, right? When you get to the PFK reaction, you're sort of committing to running glycolysis once you've made fructose 1,6 bisphosphate. So this is, as you'll see later, is a major control point in glycolysis. What's going on with PFK is a big deal for regulating how this pathway runs, right? All right, so this is the beginning of the end of phase 1 of glycolysis, right? We've now come down to the end of this process. We've made fructose 1,6 bisphosphate. From this point on, the pathway becomes fully exergonic, right? We're going to split the glucose and then get energy back, right? And we'll talk about that in the next video.